Hello, today I'm going over Cell Organelles lecture part 2. So in part 1 we covered most of the organelles. So now we have a few more that we have to go over. Not so much in matter of importance between part 1 and 2, just kind of separated. We're going to have a couple organelles that are found like only in plants here. So we're just filling in the details, really. Okay, so the first thing we're looking at is mitochondria, and we have the overall image of the cell here with a whole bunch of organelles inside, and then highlighting the mitochondria, and that shows a couple in that image. Mitochondria are bean-shaped organelles with two membranes that produce energy for the cell through chemical reactions inside their folded inner membrane. And we've seen some other organelles that have double membranes, more than just one membrane. These folds increase surface area, allowing efficient conversion of food molecules into usable energy. Unlike most organelles, mitochondria have their own DNA and ribosomes, suggesting they were once free-living prokaryotes that formed a symbiotic relationship with large cells. So these are how we get our energy, right? We eat food, and then that food can get converted into a usable form of energy by the mitochondria. All right, let's go to the next one here, and what we're looking at is the vacuole. And this is a good image. They're not always this size, but vacuoles are these large extra spaces in cells, but they have a purpose. A vacuole is a fluid-filled sac in cells that stores materials such as water, food molecules, ions, and enzymes. Plant cells have a large central vacuole that holds watery fluid to maintain the cell's strength and structure. So not only is it used for storage, but like they're saying with plants, it's also used to keep the plant rigid, among other things, obviously, but that's one of the things that helps to give it its shape, since plants don't have bones. When it loses water, the plant wilts. The vacuole can also store toxins, waste products, or pigments that give color to parts of the plant, like flower petals. So it's kind of just a separate space, and it's used for many different things. It does talk about toxins and waste products being separated from the rest of the cell in a vacuole, or other things like pigments, which are totally separate, that are found in the plants. All right, this one is lysosomes, and you can't, the image here, they're pretty small. There's just these green dots that you see in here. So they're smaller, and they're another membrane-bound organelle, but smaller than like the vacuole and stuff. Lysosomes are membrane-bound organelles that contain enzymes to defend the cell from bacteria and viruses and to break down worn-out cell parts. This is what you commonly hear lysosomes are for, breaking down old parts. They are common in animal cells and present in fewer numbers in plant cells. Lysosomes form when enzymes made in the rough ER are activated in the Golgi apparatus, allowing them to digest materials safely within their membrane without harming the rest of the cell. So it's separating stuff inside of the lysosome from the rest of the cell so it can break down. It has the tools that it needs to break stuff down inside the lysosome. But it's kind of like the vacuole, where it's separating stuff inside the vacuole from the rest of the cell. Same thing with the lysosomes. The centrosome is a region in animal cells that produces microtubules and contains two centrioles arranged at right angles. So you can see the enlarged version here. And most of the time in the cell, they're just hanging out. They have a specific purpose, but most of the time they're not doing much. Before cell division, the centrosome duplicates and microtubules form spindle fibers that help divide the DNA between two new cells. Centrioles also help organize microtubules to form cilia and flagella, which move fluids or help cells swim. The main purpose, though, is here. It's cell division. And since cells are not always going through cell division, the centrosome is not always used. Its big use comes during cell division, where it pulls apart DNA to two different cells. And there's images of that, which you'll see at another time. Okay, a couple of plant organelles, and then we're all done. So this is the cell wall, and it shows a primary wall, a secondary wall, and kind of like a middle layer. But we'll find cell walls primarily in plant cells, but not only in plant cells. Cell walls are rigid layers that surround the cell membrane in plants, algae, fungi, and most bacteria, providing protection, support, and shape. So cell walls are pretty rigid, unlike the membrane that surrounds a cell in like humans, which is pretty squishy. The cell wall is rigid for a reason. In plants and algae, they are made of cellulose, while fungal cells, cell walls contain chitin, and bacterial ones contain peptoglycan. These walls can connect between cells to support entire organisms and include channels that allow water and small molecules to pass through. So again, a big part is the fact that it provides a shape and support for the plant or for the algae or whatever it may be. But as an extra side thing, you can see little pores, little channels here. That allows material to go between cells through the cell wall. All right, the last one here is chloroplasts. And we should have chloroplasts. Looks like they're right here. There would be a whole bunch of them in the cell, just like mitochondria. Like there's a whole bunch of mitochondria. Chloroplasts are organelles that perform photosynthesis, converting sunlight into energy-rich molecules for the cell. So that's their purpose. They're going to collect the sunlight and make energy-rich molecules. Then those molecules, that'd be like food. Then that could be used by the mitochondria to harvest energy. Okay? So that's why you're finding mitochondria also in plants as well as animals. They have outer and inner membranes, and inside are thylakoids containing chlorophyll, which give plants their green color and captures light energy. Like mitochondria, chloroplasts have their own DNA and ribosomes, suggesting they were once free-living prokaryotes that formed a symbiotic relationship with larger cells. Same thing we learned about the mitochondria. It appears as if the mitochondria were free-living on their own and were enveloped or formed a symbiotic relationship with other cells. Okay, that is just the very brief introduction to these organelles. There's lots more to be learned about each one in particular, but we have to start somewhere. Hope this helps. Thank you very much.